Did you know that a single leaf can help fight deadly bacteria? Good day, everyone. Our group will present the phytochemical composition in vitro antioxidant activity and antibacterial properties of Dorstinia barniniana twine fourth leaf extract. Now, let's dive deeper into our discussion together with Group 5 members. Let's hear the brief overview from one of our group members, Hizmundo Chaga.
The study was conducted to examine how ethanol, ethyl acetate, and petroleum ether impact the extraction of phenolics, flavonoids, and the antioxidant and antibacterial properties of Yersinia barnemiana. Yersinia barnemiana emerges as a promising candidate for alternative therapies as the increase in multi-drug resistant pathogens pose significant challenges for infectious diseases. The gram-positive bacteria Staphylococcus aureus and Staphylococcus epidermidis were used in the study, along with Escherichia coli and Pseudomonas aeruginosa, which are gram-negative bacteria. The results in the study showed that ethanol extract exhibited the highest phenolic and flavonoid content, suggesting that the bioactive compounds extracted with ethanol are effective in combating pathogens. In contrast, extracts obtained using ethyl acetate and petroleum ether displayed reduced antibacterial efficacy, especially in lower concentrations. Overall, the findings of this study suggest that Yersinia barnemiana can be a source of new antimicrobial agent amidst rising antibiotic resistance. Now, let us move on to the major findings in the study to be discussed by Sexon Ricilin.
The study found that leaf extracts of Dorsiniana barmiana possess antibacterial, antioxidant, and phytochemical properties. Among the three solvent extracts tested, the ethanol extract showed the strongest antibacterial activity, producing the largest zones of inhibition, especially against Staphylococcus aureus, the most susceptible bacterium. In contrast, Pseudomonas aeruginosa was the least susceptible, possibly due to its gram-negative outer membrane that provides extra protection against antimicrobial compounds. The ethanol extract also contained the highest amount of phenolic and flavonoid compounds, which are known for their antioxidant and antibacterial properties and may explain its stronger activity. In addition, antibacterial activity increased as the concentration of the extract increased, indicating a concentration-dependent response. Overall, the findings suggested that Dorsiniana barmiana leaves may be a promising natural source of antibacterial and antioxidant compounds and could serve as a possible alternative treatment in response to growing antibiotic resistance. Now, for the interpretation of antibacterial results, the ethanol extract showed the strongest antibacterial activity because it produces the largest zones of inhibition, suggesting that ethanol extracted more active phytochemicals from Dorsiniana barmiana leaves. It also had the highest phenolic and flavonoid content, which contributed to the antibacterial and antioxidant effects. Antibacterial activity increased as the concentration of the extract increased, especially at 50 mg per milliliter, showing a concentration-dependent response. The active compounds may damage bacterial cell walls, affect membrane function, or interrupt important cellular processes. Now, let's move on to gram-positive and gram-negative to be discussed by De Castro, Jean Alexis.
Okay, moving on, let us now differentiate these two terminologies mentioned in the study. Gram-positive and gram-negative differ mainly in their cell wall structure, which affects their susceptibility to antibacterial agents. Gram-positive bacteria have a thick peptidoglycan layer, but no outer membrane, making it easier for plant compounds to enter the cell. In contrast, gram-negative bacteria have an additional outer membrane with lipopolysaccharides or LPS, which acts as a protective barrier and makes them more resistant. In the study on Yersinia barnemiana leaf extracts, gram-positive bacteria were found to be more susceptible because of these structural differences. Overall, bacterial structure plays an important role in how effectively plant compounds can affect bacterial cells. Let us now proceed on the topics of phytochemicals, which will be then explained by Ms. Wadayo Laika.
Thank you for that, Jean. For this section, we will discuss phytochemical analysis. So, what are phytochemicals? Those are natural bioactive compounds found in plants that help protect against microorganisms and environmental stress. According to Koshny and Lamb 2005, many phytochemicals possess antimicrobial properties that inhibit bacterial growth through different mechanisms. The plant extract contain phenols, flavonoids, alkaloids, terpenoids, tannins, and also saporins, glycosides, steroids, and anthraquinones. But now let us first discuss phenols. According to Lubiuk et al. 2023, phenols inhibit bacterial growth, quorum sensing, and biofilm formation, which can weaken bacterial survival. Next are flavonoids. According to Koshny and Lamb 2005, flavonoids act as a natural antimicrobial agents. Shamsuddin et al. 2022 also explained that they disrupt membrane function, inhibit nucleic acid synthesis, and reduce biofilm formation. So another important phytochemical group is alkaloids. Wang et al. 2022 stated that alkaloids interfere with bacterial proteins and enzymes, weakening bacterial growth and reproduction. Next, we have terpenoids. According to Wang et al. 2022, terpenoids disrupt bacterial membranes, causing leakage of cellular contents and affecting energy production. And lastly, we have tannins. According to Wang et al. 2024, tannins inhibit bacterial enzymes and nucleic acid synthesis, reducing bacterial growth and survival. In summary, different phytochemicals contribute to antibacterial activity through different mechanisms. According to Matotoka et al. 2023, higher concentrations of bioactive compounds are often associated with stronger antimicrobial activity. Therefore, a richer phytochemical composition results in greater antibacterial potential because multiple phytochemicals work together to inhibit bacterial growth and survival. So that concludes the phytochemical analysis. Now let us proceed to the critical evaluation of the study by Desi et al. 2026, which will be presented by Edri Janelle Macaspa.
Thank you, Ms. Laika. Let's dive into a critical evaluation of the study. First, the data clearly shows that the ethanol extract yielded the largest zone of inhibition. This proves that ethanol is an exceptionally effective solvent for extracting high concentrations of cell-disrupting phenolics and flavonoids, which is exactly what we needed to find active chemical scaffolds. Second, we see a clear link between bacterial structure and drug susceptibility. Gram-positive Staphylococcus aureus was highly susceptible because of its simpler cell wall, while gram-negative Pseudomonas aeruginosa resisted the extract due to its protective outer membrane. Also, concentration-dependent response is observed that peaked 50 mg over ml, providing that higher dosages successfully deliver more active compounds to interrupt the cellular processes. Now, while these laboratory results prove strong antibacterial efficacy, in vivo toxicities testing is recommended in order to guarantee these phytochemicals don't harm healthy human cells. Ultimately, this study successfully validates Dorstemia barnemiana as a promising new chemical template to help combat the global threat of antibiotic resistance. To discuss what this means for real-world antimicrobial resistance, let us now hear from Ms. Hassan Ranaya.
Thank you, Ms. Makasta. For real-world relevance to antimicrobial resistance, anti antibiotic resistance is caused by the overuse and misuse of antibiotics, leading to bacteria developing resistance to antimicrobial drugs. As a result, infections become more difficult to treat and contribute to increased disease spread and mortality worldwide. This had lead researchers to explore medicinal plants as alternative antimicrobial sources. For medicinal plants contain bioactive compounds such as phenolics and flavonoids that may exhibit antimicrobial activity. In the study, Yersinia barminiana ethanol extract showed high antioxidant and significant antibacterial activity, particularly and against Staphylococcus aureus, indicating its potential for developing and new antimicrobial agents. And for the scientific validation, is this is essential to ensure safety, effectiveness, and proper dosage. The study used laboratory methods such as the Foley-Chucal-2 method, DPPH assay, agar-well diffusion, and MIC and MBC determination to confirm the reliability to plant antimicrobial properties. For the risk of assuming natural means safe, natural products are not inherently safe, as some may contain toxic or harmful bioactive compounds. Incorrect dosage or improper use may lead to adverse health effects, making scientific testing unnecessary before the therapeutic application. So, therefore, both natural and synthetic antimicrobial agents must be used responsibly. Avoiding misuse and overuse helps reduce antimicrobial resistance, maintain treatment effectiveness, and protect public health. Now, let's hear it again from Ms. Makaspak.
Thank you, Ms. Rain. For the local plant, the group chose Codeum luzenicum, locally known as putak, an endemic Philippine plant traditionally used to treat stomach aches and binat, a form of postpartum or post-illness fatigue. But its true power lies in modern medicine. Researchers Ferrer, Ong and Jacinto from the University of the Philippines discovered that a fraction of this plant, called CL11, actually defeats multi-drug resistance and colon cancer cells. It works by jamming the cellular pumps that cancer cells use to spit out chemotherapy drugs. Because bacteria use similar pumps to resist antibiotics, Potak holds massive antimicrobial potential as well. However, because CL11 causes potent cells death, further scientific validation is needed to determine its toxicity, correct dosages, and long-term human safety. Unfortunately, Potak is now regionally endangered due to a habitat loss. Protecting our rainforests isn't just about conservation, it's medical necessity. And this proves that Philippine biodiversity is a place that holds the key to solving global health challenges. For the summary, let's hear again from Ms. Ranaya.
Okay. For the table summarizes the phytochemical composition, antioxidant, and antibacterial activities of Yersinia barnimiana leaf extract using ethanol, ethyl acetate, and petroleum ether solvents. Among the solvents, ethanol produced the highest extraction yield and showed the greatest phenolic and flavonoid contents. For the antioxidant activity, the ethanol extract demonstrated the highest DPPH radical scavenging activity and ferric reducing antioxidant power, indicating strong antioxidant potential. So, in terms of antibacterial activity, the ethanol extract showed the strongest inhibition, especially against Staphylococcus aureus, while petroleum ether showed the weakest effect. MIC and MBC results also confirmed that inhibitory and bactericidal activities of the extracts. So overall, the study concluded that Yersinia barnimiana, particularly the ethanol extract, is a promising and source of natural antioxidant and antibacterial compounds with potential pharmaceutical applications. And now, these are the references that we use. And that's all. Thank you for listening. Again, These are the Group 5 BS Bio 1, 2.